Awesome. Thanks so much, Stephen. Yeah, thanks to Stephen and MetaCell for hosting this workshop. And thank you to everyone for attending. Hopefully at some point we get to do these workshops in person again, but also, being virtual does give us the opportunity to have a broader reach than just, people that are local to us or can fly to a in-person workshop, which is great. So let me share my slides. So I'm going to give the first kind of overview lecture of the Miniscope project. And so this is aimed to give a bit of motivation for why we develop Miniscopes, how we've developed the Miniscopes, and why they might be a useful tool for researchers. We'll go over a little bit of the differences between our previous versions versus kind of what's the newest available systems out there. Also, I think most of what we'll talk about can generally be applied not just to our Miniscopes, right? There's a whole bunch of other open source miniature microscopes that share a lot of common design features. There's also a handful of commercially available miniature microscopes, which also share a lot of these design features. So hopefully, even if you're not interested in the UCLA Miniscope, you'll get something out of this talk and the whole workshop, because a lot of it can be broadly applied to this field in general. Again, I'll try to leave some time at the end for questions. If you have questions during the talk, type them into chat, and then we'll try to address as many as possible. And then also, as Tristan mentioned, there's breakout rooms spread out across the workshop. And so also, if you have any other questions, you can always jump into any of the breakout rooms later on in the day to ask them. Cool. So first, like super fast, somewhat accurate, somewhat not accurate kind of cliff notes of what we're trying to achieve when we image neural activity. So we can roughly think of a neuron as acting like a weighted summer. It has all these upstream neurons that get sort of summed together. Their inputs get summed together. And when the inputs reach a threshold, the neuron goes into this non-linear phase where it has this brief, about 1 millisecond wide action potential. That's a change in voltage of about 100 millivolts across a millisecond in time. There's a little bit of a refractory period where it's not able to spike again, and then it can go right back into producing action potentials. Now, this is an electrical signal which doesn't on its own produce light that we can optically see, right? So what we have to do is we have to modify these cells to be able to respond to these electrical action potentials and then produce some visible sign that they're occurring. And so for the vast majority, all miniature microscopes for the most part, and most neural imaging in head fix or fully behaving animals rely on calcium influx into the cell following an action potential to produce fluorescence. So when there's an action potential, there are channels on the membrane of the cell that open up, and calcium from outside the cell flows inside. And while an action potential is quick, a millisecond or so, the inflow of calcium into the cell takes, on the order of tens of milliseconds, something like 10 to 30 milliseconds. So it's much slower. And then it diffuses out of the cell even slower, which that diffusion can take, 100 milliseconds. And we rely on the calcium concentration in the cell. We modify the cell to produce a fluorescent protein that has been modified so that when it's not around calcium, the protein's in a physical state that's not able to fluoresce. And then when calcium is around this special fluorescent protein, it changes the structure of the fluorescent protein and enables it to fluoresce. All this really means is that as calcium concentrations increase in the cell, that cell becomes more likely to be, every single one of these fluorescent proteins in the cell becomes more capable of fluorescing, and then we're able to see that. So we are indirectly measuring neural activity by looking at the fluorescent response of the slower calcium dynamics. And then as we'll talk later on in the analysis talks, we can apply some deconvolution methods to try to go from these slow calcium dynamics to the faster underlying neural activity. Okay, so now why would we maybe want to use miniature microscopes? So there are a few kind of neural recording capabilities I've listed on the right. You might want to be able to see individual cells. You might want to record them from not just one session, but across multiple sessions. You might want to be able to do this in freely behaving animals. And so there's EFIS techniques. Hold on. In 1 second, my dog is just whining. I will really be back in 10 seconds. Okay, sorry, she wanted to be part of this talk apparently. Okay, so there are electrophysiology techniques, things like you want to record sub-threshold activity, you maybe have to patch onto a cell. Outside of a few exceptions, you really have to do this in a head fixed prep. If you wanted to record action potentials on single cell resolution, you can implant a silicon probe like what's shown on the left, or a tetrode array that oftentimes can get tens of cells in freely behaving mice. But being able to claim that the cell you see on session 1 is the same cell you see in session 2 is difficult, if not impossible, with EFIS techniques. Now, there are a bunch of optical techniques, true photon, confocal microscopy, wide field fluorescent microscopy, and outside of a few exceptions, those have to be done in head fixed animals. So you head bar an animal, you sit them under a large tabletop microscope, and you can image neural activity. You can, because you get not just functional activity, but you also get the spatial distribution of cells, you can come back day after day and identify the same cells and track the neural activity across days, weeks, or maybe even months. And over the last decade, there's been and continues to be huge improvements in these genetic reporters of neural of calcium activity, GECIs or GCAMs, that just get better, faster, and brighter, which is really great. But conveniently, there's this open spot in the middle of this table, and that's where our project comes in and a bunch of other miniature microscopes. We try to leverage all these advancements in these fluorescent reporters of neural activity getting better and brighter, but being able to do this in fully freely behaving animals. So we design wearable, lightweight miniature microscopes that can be mounted onto an animal down to the size of a mouse, and with some modifications or using other open source miniature microscopes can be worn on even smaller animals. And those animals now can freely behave, perform a bunch of behavioral tasks while we record across hundreds to maybe thousands of neurons with single cell precision, be able to track those individual neurons across days, weeks, or months while the animal's performing behavioral tasks. And just to give a bit more motivation here, now it's been a few years ago, but Nature Methods named imaging in freely behaving animals their methods of the year. And I think we're really just at the kind of beginning of what this type of technology can do. And anything that allows us to get better, more robust data out of an animal while that animal is able to more naturally behave is just a good thing for neuroscience, right? So there are experiments that just require an animal to be head fixed maybe, but whenever you can move away from that and get into a more natural behavior, generally good for science. So the way our miniature microscopes and all single photon miniature microscopes work is using something called epifluorescence or wide field fluorescent microscopy. So if we just for a second forget that we're talking about miniature microscopes and just imagine I have a slice of brain tissue and I've labeled some of these cells to have this genetic reporter of neural activity, or GCAMP is what we basically will see in all the talks. Those are those 6 cells, and I just put it under the objective of a large tabletop microscope. These fluorescent reporters don't fluoresce on their own. They need energy to fluoresce. So we provide that with blue light. So we illuminate the tissue under a microscope with a blue excitation wavelength. Now, cells that are within that blue excitation light that have been labeled with GCAMP and that are active will fluoresce green. That green light travels in all directions from the cells, but some of that fluorescence makes it back up the tissue into the optics of our microscope. And at the top of the microscope, there's really just kind of a camera. Tabletop microscopes might have a fancy EMCCD camera or scientific CMOS camera. But in the end, you just form an image of the tissue underneath your microscope. And here, an image might look like this if all 6 cells are active. We have a focal plane that sits inside the tissue. Cells on or near that focal plane will be kind of crisp and in focus. So cells 1, 2, and 3 are in focus. But because wide field or single photon fluorescent microscopy isn't able to optically section the tissue, we can't kind of define the fluorescence to only come from a thin slice in the tissue. Instead, we just kind of illuminate the whole volume underneath our microscope. And a lot of that fluorescence from the volume makes it into the microscope. We also collect fluorescence outside of our focal point. So we'll still maybe see cells 4, 5, and 6, but they'll be out of focus. And as you go deeper into the tissue, cells that fluoresce, even if your focal plane is deep in the tissue, the cells, when they fluoresce, that fluorescence has to travel up through more brain tissue, and brain tissue is a highly scattering medium. So as you go through a greater thickness of tissue, cells just look like they get more out of focus, and it's because the light is getting scattered through the tissue, and this really limits how deep we can image through a section of brain tissue to about 250 or 300 micrometers. If you try to go deeper, our microscopes are capable of imaging deeper, but if you try to, you just generally can't resolve single cell bodies because the fluorescence gets scattered too widely. You just can't resolve kind of the structure of single cells. I think for the sake of this talk, I'll skip. I have kind of the same sort of cartoon animations for different microscopy approaches that oftentimes in neuroscience, people are maybe more familiar with. So if you're not constrained to building a system that's running on the head of an animal through behaving, there's systems like light sheet microscopy, which can do single cell illumination, but illuminate just a sheet in the brain. So you remove some of this out of plane fluorescence. There's SCAPE microscopy, which is a pretty amazing technology, which is like light sheet microscopy, but the light sheet comes on an oblique angle and gets scanned through the volume. And as you scan really quickly, you can generate kind of 3D reconstructions of the tissue at near video frame rates. There's light field microscopy, and there's versions of miniature microscopes that use this as well, which we can talk about in a breakout room later. And Laura Waller tomorrow will talk, I'm sure, about a similar version of a technique that uses a similar idea. And then maybe a lot of people are familiar with fiber photometry. So this is really just a microscope with one pixel, right? So if you had a miniature microscope and you just averaged all the pixels in the camera into a single value, that's effectively what fiber photometry is. It's giving you kind of the bulk fluorescent response from a volume of activity underneath your cannula with your fiber or a GRIN lens. And then probably what most people are familiar with, if they're not using miniature microscopes yet, is to do 2-photon microscopy in maybe head fixed animals, or a little bit more and more they're becoming some wearable, freely behaving 2-photon microscopes. And here you scan an excitation light source focal point inside your tissue. And what this gives you is the ability to optically section the sample or grain you're looking at. And you can effectively collect fluorescence from just a single plane. And it's not as susceptible to scattering, so you can image deeper. So oftentimes when you think about using single photon miniature microscopes compared to like images and experiments using 2-photon microscopy, the real key things to think about is that we can't optically section with miniature microscopes. And this limits how deep we can image through tissue. And it also sort of limits our resolution and adds some background fluorescence because we're collecting background fluorescence. So you have to consider these things when you think about the types of experiments you want to do. Okay, great. And then miniature microscopes really kind of came into their own through this development of the first miniature microscope, one of the first miniature single photon microscopes from Mark Snitzer's group that was published in Nature Method in 2011. Soon after, they started this company Miniscopes that sells a commercial version of miniature microscopes. And commercial solutions are sometimes the right solution when doing research. But from our point of view, we really wanted a system that was understandable. So oftentimes commercial devices are sort of black boxes to the user, right? So you maybe can open them out of their package, get them running and do an experiment, but you might not fully understand how the device works. If you want to modify your device to really fit the experiment you want to do, that might not be possible. Additionally, the Inscopics miniature microscope is sort of prohibitively expensive for the vast majority of neuroscience labs. This technology is extremely powerful and doesn't have to be expensive. And so we really wanted to make a device that was understandable to everyone but that also like didn't break the bank, and that a lot kind of gave access to the other 95% of labs around the world that didn't have $150,000 to spend on a small microscope, but maybe have $1,000, or could try to figure out how to go through all of our open source designs and build the system or modify it themselves. And finally, not just kind of giving the tool, labs access to the tool is important, but by sharing all the designs and work we've done in an open source way, it's, I think, really enabled the development of new technologies around this platform and accelerated those developments, which has been really great. So that brings us to the Miniscope project. So roughly speaking, a Miniscope is really a head-mounted miniature microscope and a data acquisition box. And those two things are tethered together in most cases. And in our system, it uses a single flexible coax cable, which we'll talk about a little bit more later, to connect the two. And then you have a computer running our open source software, which we'll see a little bit later, or in many cases, other kind of third-party acquisition software, and that just connects over a USB cable. And then because you're doing these experiments in most of the time freely behaving animals, you want to also record behavior, and oftentimes that is done with a behavioral camera. Our software and other people's software interfaces with a bunch of different cameras, and then recently we've also developed our own affordable open source behavioral camera, which we'll see in a second. But we spend just as much time in developing these tools as really developing the resources and community to support them. So it's one thing to make an open source tool and share those designs on GitHub, but to really enable the community of users and developers, you have to spend a lot more work. So in this case, we've kind of focused on two groups: the users, which is the vast majority of neuroscientists, and developers that might want to kind of take what we've done as a foundation to build new devices. And so for users, we provide everything that's needed for a user to get off the ground, and this workshop is part of that. So we provide guides and tutorials, we have discussion boards, all through online resources. And now there are open source distribution companies that sell assembly kits to build Miniscopes, fully assembled miniature microscopes, and even some support. And then again, for developers, we really share everything we do in terms of source codes and circuit schematics, so that people can take what we've done and build upon it. We started open sourcing everything about now almost 6 years ago. Since then, I think over 18 countries have a lab that uses miniature microscopes. Probably it's closer to 800 labs have built and used miniature microscopes, and close to 3,000 Miniscopes that we know of have been built now. And we try to do these workshops. This is one of them that's virtual. And then hopefully at some point in the future, we can also do some of these small hands-on workshops. So to give an idea of just the differences, if you're already familiar with miniature microscopes, the differences of our previous generation miniature microscope compared to the newer version 4 miniature microscope. First, the similarities are that they share the same sort of data protocol. So all the devices we develop and share really rely on just a single coaxial cable to carry data and power to devices. And because all our devices share that same sort of data protocol interface, they all can run on the same data acquisition hardware. So you built a miniature microscope 5 years ago with our first data acquisition board that will support all current devices being developed now. And they all run in the same software. And generally, when we build new things, we try to make them as backwards compatible as possible. So even if you're still using version 3 Miniscopes, you can switch over to new acquisition hardware and software to enable new features. We use the same analysis, which there'll be a lot of talk about at this workshop. And then most of the surgical preps, which we'll see in the next talk, are really very similar between the two. The exception being that the version 4 Miniscope can also now image through just a cranial window prep when imaging superficial cortical regions. Some differences are the version 4 Miniscope smaller, lighter, has a much larger field of view, and has no moving parts. So in previous generation Miniscopes, if you want to adjust the focus, how deep you're imaging in the tissue, you have to get your animal and manually slide the top slider of the miniature microscope up and down. This is now all accomplished in all of our new devices with an electro-tunable lens. So now there's just a slider in software, which again, we'll see a little bit later, that lets us to adjust how deep we're imaging in the tissue. And so this is, what we're trying to achieve here. So this is actually a mouse wearing our first version of miniature microscopes when Tristan, Denise, and I were postdocs, and an animal kind of freely behaving in a small open field. And the top right just shows, it might not look great over Zoom, but it shows just the raw data stream coming out of the miniature microscope. We're imaging GCamp 6 FAST in this case, and each of those little dots is a cell body. And when that cell has a burst of action potentials, calcium concentrations increase, fluorescence increases, and we can see it. The bottom right is just like a minimally processed video. One of the steps we have to do is remove motion artifacts of the brain. So while our miniature microscopes are rigidly mounted to the skull, the brain has the consistency of jello. And when the animals are freely behaving, the brain wobbles back and forth. Usually, depends on what brain region and what size animal you're imaging. But the brain wobbles back and forth on the order of the diameter of a cell body. So 10 micrometers. So we have to correct that. And then we'd usually do a really rough delta F over F conversion to just visually be able to just kind of see kind of the activity in cells. So that's what we're seeing in the bottom right. Each one of those dots is the cell body. When it flashes, it's active. Just some features which we'll kind of outline throughout the talks on the right, but all of our miniature microscopes are fully open source. And we really work on making a system that works for researchers. Neuroscience is just very, very difficult to do. Doing neuroscience in freely behaving animals is very difficult. You don't want to have to deal with a tool that breaks in the middle of recordings. If they do, like if your mouse chews through the cable, because our systems are open source and generally labs have built the microscopes themselves, you're not afraid to just jump in and fix it in 2 minutes and get back on with your experiment. I think that setup is much better than having potentially a commercial system where if it breaks in the middle of your recording that you spent a month or two training your animals and prepping surgeries, you don't want to lose those animals in that experiment. So generally everything we've built, once you've built it once, you own that device. You feel confident you can fix it. And the small minor things that could break when it's on a freely behaving animal are oftentimes extremely easy to fix. And again, there's an online community to help you along the way. And then finally, it's just, affordable. And just because many open source miniature microscopes are affordable, doesn't necessarily mean they're any less capable than expensive commercial versions. And I would argue that our newest generation of miniature microscopes are the most capable around. Cool. We'll see this in the next talk, but generally, right, we said neurons on their own don't fluoresce, so we have to use a transgenic mouse or do a viral injection of GCamp in the brain region we want to image to get those cells to fluoresce. We then implant a lens near the tissue within usually a couple hundred micrometers because we can't image through more tissue than that. And then we come back, rigidly cement the base plate and the lens. It's also cemented to the skull. And the Miniscope snaps in and out whenever we want to record. If we opened up a Miniscope, this is the previous generation miniature microscopes. If we opened it up, you really would just see what you would expect to see in a large tabletop microscope. So there's an excitation light in a tabletop microscope. It might be like a laser or a xenon lamp that gets filtered. In our case, it's just this super bright, small excitation LED that gets defocused through a half ball lens to sort of, instead of shining all that light into a small region or field of view, tries to uniformly illuminate the tissue underneath our microscope. Passes through, this slide, that's just a cross section of our previous generation Miniscope. Passes through an optical filter which narrows the spectrum of our excitation light. Gets reflected off a dichroic mirror. A dichroic mirror will reflect certain wavelengths and pass other wavelengths. So in this case, our dichroic mirror reflects our blue excitation light down through our objective. The previous generation miniature microscopes used a GRIN lens as an objective lens, and we'll see how we've changed that for the newer generation. That illuminates the tissue underneath the microscope. GCamp is green, it will fluoresce green. That green light will travel back up through our objective, through the dichroic, goes through like another little kind of emission filter to kind of remove any backscattering blue excitation light, and then gets focused by this achromatic tube lens into an image on our image sensor. Even though our optics are really small, and they're definitely like not optimized, right? If you have a tabletop microscope and if you could open up your objective, there might be 10 to 20 optical elements just in the objective to correct for chromatic aberrations and correct for spherical distortions. In our case, we don't have most of that. But if we imaged, this resolution test slide, this zoomed up image, those red boxes are roughly the size of a cell body. We still have quite good spatial resolution. The key here is that the moment we put this in a brain, even imaging through tens of micrometers of brain tissue, what governs our spatial resolution is just the scattering through tissue. So we don't have to focus on making the most perfect optimized optical system in a lot of ways, because we lose that performance the moment we do single photon excitation imaging through tissue. And so instead we can focus on what's actually important, which is really making lightweight miniature microscopes, focusing on the tethering and the usability of these systems. We compared that to what our newer generation version 4 miniature microscopes are. They share a lot of similar features, but instead of this focus slider that was on the previous generation, we now have this electrotunable lens, which we'll see in a second. And really importantly, we've moved away from using a GRIN lens as the objective to using a stack of off-the-shelf achromatic lenses. We looked at the cross-section between the two. Again, they share a lot of similar components. The objectives, as you can see, are a bit different. Instead of the GRIN lens, we use a stack of objective lenses. And the optical path, which we'll see tomorrow for the excitation lights, important for some future work that we're currently starting now, where the excitation light goes through all the same lenses and optics that the emission green light goes through. And this gives us room to kind of shape the excitation light to be more uniform, but it also gives us some abilities to do other fun things, which we'll see later. And then the optics just form an image on an image sensor. The newer generations between the image sensor and the more optimized optics are somewhere between 10 to 15 times more sensitive to previous generation miniature microscopes. Because we've changed the objective side of the miniature microscope from a past generation to our newer one, what this has given us, along with more flexibility and better kind of efficiency of collecting light, is we were able to push the working distance, kind of where the focal plane sits below the bottom of the miniscope, further. So previous generations and most other open source and commercial miniature microscopes still rely on this GRIN lens objective. And this really puts the focal plane of what you're imaging to just like a couple hundred micrometers below the microscope. While this can work in many cases, if you want to image cortex, the solution was that you had to really implant this objective lens on top of cortex to be able to reach, layer 2, 3 or something. These newer generation miniature microscopes can have a much larger working distance, which lets you do the same prep you would do for like 2 photon imaging in cortex, which is just craniotomy and cranial window, and they can reach through all the glass of the cranial window and reach into cortex, which is really nice. I think for the sake of this talk, I'll skip over the kind of theory behind using GRIN lenses, but I'll just hit on the topic of you might want to image superficial layers, which is great, but the brain is a volume, and there's many brain regions under the surface. And so miniature microscopes can generally image from almost any brain region. And the way they can do this is by implanting an additional lens, which is usually called a relay GRIN lens. So if just took like a generic microscope, you have an objective that's focusing tube lens and then an image sensor. And here we're kind of simulating 4 colored cells, a blue, green, orange, and red cell. And you could just kind of put those cells on the focal plane of your microscope, and those get focused onto your image sensor. But again, you can't go deep, more than a few hundred micrometers into the tissue that way. If you want to go deeper, and we'll see the surgeries of how to do this, you can implant what's called a relay GRIN lens, and you put the bottom of that GRIN lens to within a few hundred micrometers of the tissue you want to image. And that relay GRIN lens just relays the kind of the volume of light underneath the lens and refocuses it right above the lens. So you get an intermediate image of your tissue that sits right above your relay GRIN lens. And then your microscope, a miniature microscope or a tabletop microscope, even 2 photon microscopes, then image that intermediate image. This effectively lets you get down to deep brain structures, reconstruct that volume of the brain right above your GRIN lens, and then image that. We'll skip over this. If people have questions about how GRIN lenses actually work, we can talk at a breakout room. The one key I'll point out is not all GRIN lenses are made equal. Most GRIN lenses out in the world have been developed not for this type of imaging, but have been developed to couple fibers together for telecommunication. And almost all GRIN lenses do not work well when imaging in these types of configurations used for miniature microscopy. And so we generally recommend people purchasing GRIN lenses from GRIN Tech, and Inscopics is one of the exclusive distributors of these lenses. So any relay lens in Inscopic sells is fully compatible with miniature microscopes and our Miniscopes. And also Go Photon sells a specific product line, I think they call them the CHL lenses, that are compatible with this type of imaging. But we can talk in the breakout room if you have specific questions. Okay, so what's really key when doing experiments with a head-mounted device, it turns out to not be the weight of the device. There's some, as the weight increases on an animal's head, that will affect their behavior, absolutely. But what ends up being more important than just the weight is the tethering that comes off the animal's head. And so our first miniature microscope used like a custom braided 8 or 10 wire cable. And then we quickly realized that we could effectively pack data, which usually would take 2 wires, some type of configuration communication lines, which we use this thing called I2C communication that takes 2 lines, and then a bunch of data that might be 15 lines, as well as some other control channels. You could do these as all separate lines. We did that in the past. Almost all, I would say all miniature microscopes that don't use our data acquisition box still do some version of this, which just results in a custom cable that's not flexible and can drastically impact behavior. But what we've done within the first 2 years of this project was figure out how to pack all these lines into a single flexible coax cable. So this is, if you're not familiar with what a coax cable is, it's just a single conductor with some insulation around it, and then a thin kind of grounded shield. We generally, the standard coax cable diameters are about 1 millimeter in diameter, but our systems have been tested down to a 0.3 millimeter diameter coax cable. So it's effectively, like dental floss running off the animal's head. And this is critically important for a lot of behaviors. So to support this, our microscopes end up being about 0.5 gram heavier because of the added electronics, but you gain so much from going to these thin, flexible cables. I'll just finish by talking about the other kind of components of the general Miniscope system, is that for the wired systems, we need this data acquisition box. On one side of the system, it connects over this thin coax cable between the data acquisition box and the head-mounted microscope. And then on the other side, it's just a USB connection that connects to the computer. The same any version of our acquisition boards support all versions of miniature microscopes that have been developed. And what's nice is that it turns out that this coax cable ends up being a really good idea. So that's been incorporated into the new generation of open EFIS head stages, and many other open source kind of developments of miniature microscopes end up using this combination of coax cable and our data acquisition box, which is really nice. And then on the computer side, we will go over the acquisition software we have. This we'll also see in a bit that the software embeds Python and can support deep lab cut live. So in real time, we can run deep lab cut. This is my son in real time tracking his pose through deep lab cut live. And then software works with a lot of any webcam, but we've also developed kind of a affordable, very usable behavioral camera that's open source as well. If we put everything together, we end up getting something that looks like this. This is a mouse wearing a miniature microscope running back and forth on a 2-meter long linear track. We're imaging just green fluorescence, so kind of monochromatic light coming out of the system. But once we run through our analysis pipeline, either Minion or Hyman or another pipeline, we can identify individual cells and attribute fluorescence to those cells. So then I've just pseudo-colored the activity here. There's something like 500 or 600 active neurons, and a couple of the calcium traces shown on the right. So I think with that, I will skip over this stuff because we'll talk about it later. just other imaging configurations with their new generation miniature microscope. Okay, I'll finish here. So what's really important for a lot of experiments is not just to record a neuron for one day and then come back the next session and not be sure if you're recording the same neuron. We really would like to do longitudinal recording across days or weeks or months. And imaging techniques kind of enable the ability to do this. So this is an example of imaging the same animal, across a month. So this showing the mean frame, sort of this maximum projection image that kind of shows where the cells are. Then once you run through the pipeline identifying individual cells, we can then use some pretty simple criteria to say that a cell that showed up in day 1, is that or is that not a cell that showed up near it in day 14 or 30. This allows us to really do longitudinal studies looking at learning and memory for aging, and really look at the change in neural dynamics of individual cells across hundreds or maybe thousands of neurons, as well as in free behaving animals, which is a really powerful technique. And then Tristan showed this, but I'll end here that while, if you didn't take any notes and you never watched the videos being recorded this workshop, and all you left with is kind of this feeling like, yeah, like I think I can kind of understand what's going on and I could jump in and be successful building miniature microscopes. We have all these online resources to support that. So the main kind of central website is miniscope.org, which now for all the newer developments kind of just links to these GitHub repositories. But we also have a very active Miniscope Google group that there's already a ton of excellent questions and answers there. But if you post a question, someone from the community or me or someone from the lab will generally get back to you within a few days. And then all the designs are now housed on GitHub repositories, and each of the tools, the hardware, the software, the firmware has its own GitHub wiki, which links through miniscope.org and links all together, which outlines all these tutorials and guides to really get you up and running with these tools. So I think for that, I will end here. And if there's questions for the next few minutes, I'd be happy to answer some of them. So let me stop sharing. I think Tristan was, I don't know if you were watching any of the.
I was trying to go through the chat as fast as I could. If anyone wants to ask a question, they can go ahead and raise their hand, or you can put it in the chat. There were a few questions about GRIN lenses and like the types of GRIN lenses and where to get them and the compatibility. I think Inscopics sells these GRIN lenses that are relay lenses, but they have these caps on the top. And so those are directly built for the Inscopics system that puts a GRIN lens right on top of it. And it's made to fit perfectly with their GRIN lens system. I wouldn't generally recommend those capped versions. We typically use the bare lenses, which they will also sell just without the cap. And then you don't have to worry about compatibility between the two. One thing to remember about V3 versus V4. Now, there's a lot of differences there, but the key difference is that the objective lens, the large GRIN lens in V3, in V3, it can be implanted into the animal or it can go into the Miniscope. And we'll talk about this in the surgery lecture as well. Whereas V4, it's built into the Miniscope. And so you really have to use either a relay lens or a cranial window to access your cells. We'll go over that a little bit as well.
Yeah, I see there's a question about commutators. So all the miniature microscopes we've developed that all rely on the single coax cable can go through a commutator. A coax cable is really just a conductor and a ground. So it needs a kind of a 2-channel commutator, a single channel and grounded commutator. We have in the past oftentimes used these low torque commutators from Dragonfly, which in general work well, but I think they're really hard to source right now. And if you go onto the Google group and just search for commutators, there are a few people that have come up with kind of like DIY versions where you end up buying, like some version of like a $30 commutator and slightly modify it to work well with the coax cable connection. The one caveat here is that we are, for the most part, kind of pushing the type of data we are pushing through the coax cable isn't exactly the best match for most of these kind of low torque commutators. And so we've seen in the past that if you build 5 commutators, like do-it-yourself commutators. Some will perform perfectly for a year, and some will just end up having kind of unstable data connection. So it's oftentimes a good idea to build a few of them and then test them to see which one, which one of the ones you built is reliable or not. And usually, once one seems to be reliable, it's reliable for a long time. Also, I've heard that Doric sells an optical and electrical commutator that I think works well with this Miniscopes. And I was going to say one other commutator. I know there's a bunch of additional information on the Google group. Yeah, I don't know if there's any other questions.
So what's the field of view on the V4 real quick?
So yeah, and what I'll say, so we talked a bit about the version 3 and the version 4. If you're starting with Miniscopes now, you would never get a version 3. So really, the version 4 is substantially better and kind of at roughly the same cost if you source it from open source distributors. And so you would really start with the version 4. The field of view for the standard optics of the version 4 gives you 1 millimeter in diameter. So oftentimes, if you're going a little deep, you will implant a 1 millimeter GRIN lens. If you do that, you image effectively the entire lens. If you implant a thinner GRIN lens, you'll see a small, you'll see that GRIN lens within your field of view. And then because the Miniscopes are customizable and open source, we have a guide on our wiki where you can swap out some of the lenses to change different optical parameters of the system. So you can increase your working distance or increase your field of view by just swapping out a lens. We have guides for that. So you can actually kind of push the Miniscopes, the version 4 Miniscope, to have almost a 2 millimeter diameter field of view and a bit longer of a working distance, somewhere between like 2 or 3 millimeter working distance.
Okay, we're going to have to move on.
Okay.
So Daniel, can you go ahead and answer the questions in the chat now? And then we're going to have to move on, but we also have breakout rooms where we can chat about this more. But we're going to move on to the surgical techniques right now.
Excellent.